So the Walter Reed Army Institute of Research and many different groups working within the Walter Reed Army Institute of Research joined the national effort to develop a countermeasure, and specifically a vaccine countermeasure, for the Zika virus for a number of reasons, not the least of which is that the Institute since 1893 has been working on the kinds of viruses called flaviviruses that Zika represents. We began to understand that we could develop a whole-killed vaccine, we call it a purified inactivated vaccine, fairly easily. Critically, at the same time we were even conceiving of this, because of my work with investigators at Harvard University, and most notably Dan Baruch, he and I have been working on vaccines for HIV for almost a decade. Dan called me about 3 or 4 weeks after we began to conceive of this idea, saying that he was working on Zika. He had in fact developed an animal model using mice that seemed to be promising, and wondered if I knew of anyone that was developing a vaccine. So that very quickly turned into a collaboration. So we were very excited to very early on gather very clear information from the experiments done in mice that the vaccine that both was produced in DNA by Harvard and the purified inactivated vaccine, or PIV, that was produced by WRAIR, both proved to be very effective when mice were immunized in groups of 10. We could then expose them to a challenge with Zika viruses, 2 different types, one from Puerto Rico and one from Brazil. And in both cases, the animals that were given this vaccine were completely protected from Zika infection.
I think there are a few reasons why WRAIR has been able to move so quickly in developing this Zika vaccine candidate. The first is that we have a lot of subject matter expertise. We've been working on flaviviruses for over a century. The second is that we have a full spectrum of capabilities within our research enterprise. We have the ability to do biosurveillance at our overseas laboratories. We have the ability to do basic science. And then we have the ability to actually produce and manufacture our own vaccines at a small scale so that we can support early human testing. And then I think the third and very important is that we have a network of very accomplished collaborators, both within the United States government as well as within academia.
We expect by October of this year, working with collaborators at the National Institutes of Allergy and Infectious Diseases, to do a series of initial studies in humans to make sure, first and foremost, the vaccine is safe, and then eventually move into larger phase 2 studies where we'll attempt to show that the vaccine is protective in terms of natural exposure to Zika in areas of the world now where Zika is raging.